Alright, everybody, welcome back to another lecture video. This one is on specific catabolic pathways. We're going to look at how some more details of how carbohydrates enter into the citric acid cycle, which we explored last time. We're going to look at how lipids and proteins also work their way into these metabolic pathways. So we'll start with the convergence. So you can see here carbohydrates at the top, working its way down through glycolysis, which we're going to go over. Glycolysis will eventually get you to pyruvate and acetyl-CoA, which we saw entering the citric acid cycle in the last chapter. Now, taking a step back, we can see that lipids over here on the side, that these lipids can actually also be converted into either glycerol. We know that our fatty acids contain a glycerol backbone, so to speak, or our phospholipids as well. Glycerol can be turned into pyruvate. And we can see that lipids can also be broken down into fatty acids, which can undergo beta oxidation. We'll see that means 2 carbons at a time, they can be kind of broken apart. And that'll form acetyl-CoA, which will also enter our citric acid cycle. Proteins can break down via 2 essential pathways, or 2 essential categories of amino acids. Glucogenic, which can be turned into or break down products down into pyruvate, which can ultimately be turned back into glucose if we needed to, or ketogenic, and these break down into acetyl-CoA or a few other of the intermediates of the citric acid cycle, as well as amino acids get the ammonia removed through the urea cycle, and we'll take a look at how that works. Okay. So glycolysis is where we're starting. Glycolysis is where we take that 6-carbon glucose and we just run it through 10 reactions. Now the first 5 reactions can be thought of as the prepping stage, the sort of activation stage where we have to put energy in to get this glucose into the right shape or state so that it can then be hydrolyzed into 2 smaller molecules and generate some energy. And so the first 5 reactions are prepping it. The second 5 reactions are what we call the payoff stage. During glycolysis, we're going to net 2 ATPs. What that means is we're going to end up with 2 more ATPs than we had to put in. So if we put in 2, we're going to get 4 back. The 2 that we got back canceled out the 2 we put in, but we also got 2 extra, 2 net. So reaction 1. Phosphorylation of alpha-D-glucose. So this involves a kinase. Kinases are those proteins that transfer phosphate groups, remember? So glucose gets sort of activated by addition of a phosphate group. And so now we've got alpha-D-glucose 6-phosphate. Now glucose 6-phosphate that's also the first step when we want to make glycogen, which is a storage molecule for glucose. So this reaction happens everywhere in your body. Isomerization is the second step. We're going to take alpha-D-glucose 6-phosphate and turn it into fructose 6-phosphate. This is also a reversible reaction. If we have fructose molecules, they can be turned into glucose so that we can store them if we needed to. An isomerase does this. Remember, isomerases are those that just change the form, not the molecular formula. So now we have glucose turning into fructose. And so here you can see how that sort of happens. This is called a keto-enol tautomerism. And it happens when they're in their open chain forms. And so we see the double bond forms here. We get some shifting around of some hydrogens. There was a hydrogen here that moves over here. So then now that carbon's got both those. This one's missing one. Double bond is present. Double bond moves, hydrogen moves as well. This hydrogen will go over here. Okay, so an enzyme does this. Reaction 6. This is the reaction that you have to, that basically commits glucose to entering the citric acid cycle. There's no going back after doing this. So this costs ATP. We're going to add another phosphate group, and now we have fructose 1,6-bisphosphate. So now we are committed to glycolysis. It's a high energy molecule. This one we're going to break down. So let's see. Cleavage of fructose 1,6-bisphosphate to triose phosphates, triose being 3 carbon sugars, right? So we get 2 of them. Aldolase cuts this thing in half. We get dihydroxyacetone phosphate. We're going to see that this is an entrance point later on in our one of our other metabolisms, and or I should say catabolisms, because that's breaking down. Anabolism is building up. And we get a D-glyceraldehyde 3-phosphate. That's the molecule that essentially we want. So that was reaction 4. Reaction 5 is going to deal with that dihydroxyacetone phosphate. We're going to turn it into another molecule of D-glyceraldehyde 3-phosphate. So we'll have 2 of those now. We formed one already. This was its twin, but it just looked different, so now we isomerize it. And that is done by phosphotriose isomerase. So that was reaction 5. Payoff stage now. Reaction 6. Reaction 6 starts with taking, now we'll just discuss each reaction one time, but realize it's happening twice, because now we have 2 of these molecules. So this is happening twice, because we have 2 molecules going through this. So glyceraldehyde 3-phosphate, in a series of reactions here, gets oxidized, meaning it's going to transfer electrons to NAD+ and a hydrogen. NAD+ is going to become NADH. Remember from the citric acid cycle, this is what goes down to make more energy from the mitochondria. Okay, so we've generated one of those. We're also going to add a phosphate on here. So we're going to add this phosphate to our molecule. So now we're going to have 1,3-bisphosphate phosphoglycerate. So there's 2 phosphate groups on here now. 1, 2. This is the high energy molecule that we have been working toward. The hydrolysis of this molecule is going to yield lots of energy. So 1,3-bisphosphoglycerate now, in the presence of phosphoglycerate kinase, is going to take one of those phosphate groups and transfer it to an ADP, generating an ATP. So we're going to get our first of the high energy molecules here. Okay, reaction 8 is going to start with the 3-phosphoglycerate, which is what we have now, and we want to move that phosphate group to a different carbon. So we're going to isomerize it. 3-phosphoglycerate becomes 2-phosphoglycerate. So now we're going to make this phosphate group fall off a little easier, and to do that we need to introduce a double bond here to the carbon that it's attached to. And so reaction 9 actually removes a water molecule in a dehydration reaction. So 2-phosphoglycerate becomes phosphoenol pyruvate. This is the energy molecule here that can make ATP. So phosphoenol pyruvate is now ready for reaction 10 to generate some more ATP. And phosphoenol pyruvate will transfer that phosphate group to another ADP, making an ATP and giving us pyruvate. Pyruvate is the molecule that ultimately we're going to see enter the citric acid cycle as acetyl-CoA. And I think we looked at that reaction in the last chapter. So let's kind of see how much energy we get for this. So in this process, we're going to get some pyruvates, 2 of them. We're going to get some electron carriers, and we're going to make 2 ATPs on the spot. Okay, so we didn't go over this last time, so we'll go over it now. So pyruvate, if you've got it available, oxygen available, you're going to want to take that into the citric acid cycle. So you're going to want to go this way. But some organisms, and even us and some of tissues, don't have oxygen, like in our overworked muscles, right? There's an oxygen. It's anaerobic, meaning no oxygen. So in that case, we need to figure out what to do with our NADHs. We need to reduce them, we need to oxidize them back into NAD+, or else this whole process shuts down, because you have to have NAD+ really early on in those glycolysis reactions. So let's look at what our muscles do first. Our muscles will then take pyruvate and reduce pyruvate into lactate in order to get back our NAD+. So NADH would also be entering into this process. So we make lactic acid in our muscles, or lactate, no big deal. Lactate can be taken back and turned into pyruvate and then enter into the citric acid cycle, or made into glucose if we needed to. Lactate's okay for us. Yeast, if it doesn't have any other options, will start to make ethanol. Now, yeast can live in aerobic conditions as well, and that's when they do normal citric acid cycle. But if they're in anaerobic conditions, like when you're brewing beer or under the dirt or in other weird places, then they actually do fermentation. And they take pyruvate, they remove a carbon from it initially in a decarboxylase, and then an alcohol dehydrogenase reduces that product into ethanol. And so there's, I think, two reactions that happen there. But ethanol is the product. And again, the purpose of that is to get that NAD plus again so that it can go do its job. Okay, if air is present, then we're going to make acetyl-CoA. So let's see, I think we're going to see a little more details here. We can kind of go through this quickly. This is in our muscles. Lactate dehydrogenase will go back and forth, making pyruvate or lactate as needed, and reducing or oxidizing as needed as well. Yeah, so this is just showing us the overall muscle, the overall reaction. Glucose can make lactate in our muscles. Blood lactate reaches certain levels, muscle tissues become almost completely exhausted. There was a recent paper I saw maybe in the last year, maybe it isn't that recent, that was actually looking into whether or not lactate is what causes muscle fatigue. And in mice, where they actually blocked the production of lactic acid in the muscles, those mice tired out sooner. So maybe the lactic acid that's building up is somehow allowing the muscles to keep going longer when they would have normally tired out. I don't know. That's what the paper suggested. It was a neat paper. Okay. In yeast, right, we can get ethanol. Here we go. Pyruvate decarboxylase removes the CO2. This is what provides the bubble in beer and in sparkling wines, champagne. And then the second reaction, alcohol dehydrogenase, takes this, which I believe is acetaldehyde, and reduces it into that primary alcohol. So these are all reactions that we went over earlier on in the chapters, coming back to make sense for us during this troubling citric acid cycle. Pyruvate to acetyl-CoA now is that other reaction that has to happen to bridge this chapter with our last chapter. Pyruvate will also undergo a decarboxylation to make a CO2 that we breathe out. This is why last chapter we were looking at acetyl-CoA and it only had 2 carbons in it. And those were the 2 carbons that we were saying came from the sugar, or from the glucose. Well, here they are. These pyruvates were only 3 carbons. There's 2 of them because our glucose got broken in half, remember? And then those get breathed out. 2 carbons, 1 per pyruvate. And we have 2 acetyl-CoAs. We also generated an NADH, an electron carrier, and ultimately that makes like 5 or 6 ATPs down the line. Let's talk about another pathway that happens here for glucose 6-phosphate. Glucose 6-phosphate can actually be turned into ribulose 5-phosphate. Now, a ribose sugar is important because those are the starting sugars for our DNA and RNA. And if we want to make DNA, we need RNA. And if we want RNA, we need riboses. And so that's what this reaction will do. Now, ribose 5-phosphate, ultimately the molecule we get, we can still actually turn that back into glyceraldehyde 3-phosphate if we wanted to. And glyceraldehyde 3-phosphate, if you remember from a few minutes ago, was the molecule that we got after we cut our glucose in half. Our, what was it, our 1,6 fructose bisphosphate. When it got cut in half, we got a glyceraldehyde 3-phosphate. So it can enter into that pathway if we needed it to. So, totaling up from this chapter and last chapter, it cost us some ATPs initially to add on to the glucose to glucose 6-phosphate, and then the second one to get glucose fructose 1,6 bisphosphate. 2 ATPs, but in the end we ended up getting 4 back from glycolysis. We got some more back when we got some back from glycolysis here. We got some more back from the NADHs and what they were able to get from the electron transport chain. We got another couple of NADHs from the change of pyruvate into acetyl-CoA, and then in the oxidation of the acetyl-CoAs via citric acid cycle, we generated something like 6 NADHs, 2 FADH2s, 2 GTPs, and I think that's it. So adding all that up, we get 36 or so ATPs per glucose molecule. That's not too shabby. Now, for comparison, let's look at how much we can get actually through some of the other pathways. Let's see. Oh, okay, so this is fatty acids. So fatty acids, remember, are made up of a glycerol backbone, or a glycerol and some fatty acids. The reaction, right, between the alcohol, glycerol, and the fatty acids, or the carboxylic acid, to make those esters. So glycerol can enter right into glycolysis via dihydroxyacetone phosphate. This was the other twin when fructose 1,6-bisphosphate got broken in half. We got 3 hydroxy phosphate. No, what was it? 3 glyceraldehyde, 3-phosphate. Excuse me. We were just talking about it. We got glyceraldehyde 6-phosphate, and this guy. And this guy had to be turned around or isomerized. I think it was an enolase or something that did it. So this can enter right into, I think this guy was reaction 5 of glycolysis. So glycerol can go right into glycolysis and be turned into carbon dioxide and energy. Now the fatty acids, on the other hand, those go through a different method of breaking them down. And just for comparison, a glucose will give us, what did we say it was, about 36 ATPs? Let's say that energy is about 3.8 kilocalories. Well, palmitic acid will give us about 9.3 kilocalories per gram. I guess per mole it even looks like a much bigger number, but grams are, a little more you can picture a gram a little better. A dollar bill, the weight of a bill, any paper money, is 1 gram. So 3.8 is like almost $4 if you were holding it, right? That much sugar gives you about 600, or well, what do we say? 3.8 kilocalories. Anyways, the point I'm making here is that the fatty acid is more, way more. So let's talk about how we get into the citric acid cycle as a fat. So beta oxidation goes through 5 reactions. We're going to look at those very quickly. But we're going to see that essentially we take 2 carbons at a time from the carboxyl end, and so that's this end. Right? This is the fatty acid side. So we're going to take them off of here. Sorry. It's going to be this carbon and this carbon. We're going to take those 2 at a time until all of our carbons are gone. And remember, most of the carbons in our bodies are even numbered because we make them that way. That's how we can break them apart completely. So the first reaction of a fatty acid is actually going to require some ATP and an acetyl-CoA molecule, and we're going to form this acyl-CoA. So the fatty acid is right here, and we formed a bond with the CoA, and that cost us some energy. Now the second reaction is actually going to generate an electron carrier. We're going to do an oxidation reduction. FAD becomes FADH, and we get oxidized into a trans-enolase. So what happens is we lose some hydrogens and the formation of a double bond. The alpha-beta carbon-carbon single bond becomes double. Reaction 3 is going to involve turning our trans-enoyl CoA into a primary alcohol. And so we're going to basically form this group here on this carbon right here. This is a hydration, so we're just adding OH and H back to these 2 carbons. Reaction 4 is going to oxidize that secondary, sorry, did I say primary alcohol? It's going to oxidize that secondary alcohol to a ketone. And so now we're going to have this neat little molecule that has 2 carbonyls. And so we can see now that if we were to remove these, it would almost look like we still have the rest of the fatty acid. We've essentially regenerated the rest of it, and we've got the end on it and everything, ready to go and react with another coenzyme A, when we get there. So that's essentially what's going to happen. We've just primed it for that. And so this was done by beta-hydroxyacyl-CoA dehydrogenase, and another high-energy molecule was made. Electron carrier. So in addition to glycolysis making ATP and NADH and FADH2, and in addition to getting these bigger molecules like these fatty acids broken down into little bits that we can get into glycolysis or the citric acid cycle, these processes themselves are also generating electron carriers. So this is where all that extra energy comes from. So reaction 5 is taking that, or basically cleaving off the rest of the fatty acid from the 2 bits that are going to get stuck to coenzyme A. So we've got cleavage of the carbon-carbon chain by a molecule of coenzyme A. So another coenzyme A actually comes in here and helps cleave this by this guy reacting with, I guess, that R carbon, so that this guy can break off. And this is just exactly what we would have gotten if pyruvate had been oxidized, given off a CO2, and added to a regular molecule of coenzyme A. We would have had acetyl-CoA. So, and then this is another guy. This is another, effectively 2 carbons shorter molecule that can undergo those 5 reactions again. So this is called beta oxidation, and it's how we get fatty acids broken all the way down into something that can enter the citric acid cycle. Okay, so depending on how many carbons you start with, this repeats over and over and over. So you can see here lots of NADs, lots of FADs, 8 cycles of beta oxidation will ultimately wrap this up. There's an extra step if you have unsaturated fats where they have to turn cis double bonds into trans double bonds. I don't think we showed that reaction here. So 36 for glycolysis, 36 for glucose metabolism or carbohydrate metabolism, 146 for fatty acids. So per mole of stearic acid, and this isn't even counting any kind of glycerol, right? Glycerol entered into glycolysis at a different step. This is just the fatty acid part. Stearic has 18 carbons, 2 to prime it, and then all of those given off during each of those steps, all those FADHs and NADH2s. So that's fatty acid metabolism. Now ketone bodies are something else that happens. Ketone bodies are acetone, beta-hydroxybutyrate, and acetoacetate. These can be used for energy. They can be broken down, or they can be turned into things that will enter those same pathways. Acetone, for example, can be added to coenzyme A, and you have acetyl-CoA. This happens when you have too much acetyl-CoA relative to the amount of oxaloacetate. Acetyl-CoA has to react with oxaloacetate to form citrate in the first reaction of the citric acid cycle. So if you don't have any oxaloacetate available, this sort of just hangs around and builds up a little bit. Reasons for that? Well, if you are too high in lipids and too low in carbohydrates, if you're on that kind of keto diet, diabetes can be a factor. Starvation is typically when you are not eating any carbs and you're just burning your fats and eating too many fats. So all of these things are kind of related to the formation of ketone bodies. These are them. So you can see acetyl-CoA can be converted into acetoacetyl-CoA, which then can generate some energy via the electron carriers, forming beta-hydroxybutyrate, or we can breathe out CO2s and have acetone. I don't think your book goes into a lot more detail on where these products fit back into the cycle, but these are some of the products. Big picture of protein sort of entrance into the pathways here. So proteins can be hydrolyzed into amino acids. Amino acids can either be used to make more proteins. They can be used to make the things that are similar to or the amino components of things like heme or nucleotides, right? Because we have nitrogenous bases. We've got hormones and neurotransmitters that often have nitrogens or amide or amide functionalities, and so the aminos are the source of nitrogen, and so those go to synthesize many of those other compounds. If we're not going to make something with them, then we're going to break them down, and that's oxidative deamination. That means removing the amino groups. This is done pretty easily, and then amino or ammonia, or ammonium, I should say, if in that ion form, NH4, enters the urea cycle to be excreted. Once your protein or your amino acids don't have nitrogens on them, then we have the carbon skeleton. This can be broken down into pyruvate or to acetyl-CoA, which can be broken down via the citric acid cycle. Pyruvate itself can either be made into more glucose if we needed to, so protein into glucose, or pyruvate can be, again, sent into the citric acid cycle. And then we also know that the fats can enter in here as well. So this is like an overview of that pathway. Now let's focus in a little bit on the nitrogen. So first is that amino removal, transamination. This is done by a molecule called pyridoxal phosphate. So pyridoxal phosphate and the amino acid in question form something called an imine. This is where we have the double bond between this N and C. An enzyme comes in here and isomerizes this from being from one carbon to the other carbon, still an imine, and then this basically hydrolyzes that bond and cuts it, and so the nitrogen just got switched from this guy to this guy. And then pyridoxal phosphate will go and transfer that amino group to something else. Let's see here. Nitrogen to be excreted is collected in glutamate. So glutamate is where the nitrogen ultimately ends up on its way to the citric acid, to the urea cycle. Glutamate gets turned into alpha ketoglutarate, which is the removal of this ammonium ion. This generates some energy in the form of an electron carrier. Here's the sort of big picture of the urea cycle. So you see here the ammonium entering at the top. Carbon dioxide and ammonium combine to make carbamoyl phosphatate. This requires a phosphate group from ATP, so there's some energy cost here. Carbamoyl phosphatate can enter the urea cycle. And so in this urea cycle, it can accept amino groups from aspartate, turning aspartate into fumarate. Fumarate, of course, being part of the citric acid cycle. There's some other reactions in here. We're going to look at them. Urea, ultimately the recipient of the amino groups coming from aspartate, and the carbamoyl group coming from that extra ammonia and carbon dioxide. So look at all the stuff we want to get rid of. We got a carbon dioxide we want to get rid of, and we've got two nitrogens we want to get rid of. Urea is beautiful. Okay, just a couple of the more details of that reaction. We don't need to focus on this. Again, big picture is we've added two nitrogens onto this molecule, and we're gonna pee it out. Okay. So amino acids can be broken down, if we're not going to use them, via two categories: the glucogenic kind or the ketogenic kind. The glucogenic ones break down into pyruvate or oxaloacetate, and both of those can be turned back into glucose if we need to, or they can go through the citric acid cycle. Ketogenic amino acids get degraded into acetyl-CoA or acetoacetyl-CoA, and we saw both of those were tied to those ketone bodies, or they can get turned into acetone or whatever the other ketone body was. Here is a nice picture of that. You can see different groups. So we've got the ketogenic amino acids here in kind of beige, and we've got the glucogenic amino acids in purple. They enter into different areas of the citric acid cycle. Again, pyruvate can be turned into glucose, pyruvate can be turned into oxaloacetate and then into glucose. Asparagine, tyrosine, isoleucine, glutamate, they can all be turned into intermediates of the citric acid cycle that can all be turned into oxaloacetate and turned into glucose. And so that's why they're glucogenic. If we needed glucose, which is what our body runs on, we can use proteins to get it. Very neat. Here's that classifications and categories of the different amino acids and which metabolism they fall under, or catabolism. Red blood cells. So heme catabolism. Heme is the iron holding part of or our oxygen binding part of our protein hemoglobin. The globin is hydrolyzed into amino acids. These are the protein chains, and reused. The iron is saved in something called ferritin, stored and reused, and the heme is turned into bilirubin. And that enters the bloodstream, stored in the gallbladder, stored in the bile, excreted in the feces. So heme looks like this, and bilirubin looks like this. And that is the breakdown of that red blood cells. And this, I believe, is the last slide, that we need to look at. This is basically showing everything, where it happens and how it happens. So this is a mitochondria, site of the citric acid cycle. And we see just the various amino acids entering, and then how they, either are they formed in the cytosol, do they have to get transported in to the mitochondria or not? Okay. So I hope this chapter wasn't too overwhelming. We did go a little fast. A lot of what we talked about in the citric acid cycle from the last chapter, I would imagine you would still know. So this kind of fills in those gaps, glycolysis and how protein and fat synthesis or breakdown, factor in, and how they all enter into that citric acid cycle and ultimately into that electron transport chain, where we looked at those complexes and how ATP got synthesized. Okay, well, I think we only have one more lecture after this. I'll have to double check my list, but hope you guys are staying well, and I will see you next time.